Hi everyone, my name is Rridhisha Kumar and I'm from the Suzuki Lab at the J. Craig Venter Institute, and today I'll be talking to you about one of our projects, which is the insertion of a minimized carbon fixation pathway into the JCVI minimal cell. So our motivation for this project lies in this graph here, where you can see that the rapid increase in carbon dioxide emissions is reaching highly unsustainable levels to the point of major damage. Because of this, researchers have been focusing on finding ways to not only reduce emissions, but also develop systems of carbon fixation so that we can mitigate the effects of climate change. Over here in white and yellow, you can see 6 naturally occurring carbon fixation pathways, where the most common one is the Calvin cycle. Majority of the atmospheric carbon dioxide is fixated through the Calvin cycle, which is predominantly found in plants and algae. However, it is limited by its high ATP cost and its rate-limiting enzyme rubisco. Additionally, a common feature of these naturally occurring cycles is that they have a high number of reactions, which makes it harder to optimize in vivo. Because of that, we wanted to focus on synthetic carbon fixation cycles that have been created to be more consolidated and more efficient for fixation. One of these is the POAP pathway, which has a higher carbon fixation rate than the Ketch cycle, which was the first synthetic cycle, and it can fixate carbon at a rate of 8 nanomoles of carbon dioxide per minute per milligram, and it only has a quarter of the reactions and enzymes needed, which is just 4. So looking more into this pathway, it starts with acetyl-CoA, which is converted into pyruvate using the enzyme pyruvate synthase, or P4. This is then converted to oxaloacetate using pyruvate carboxylase, or PYC. Oxaloacetate is broken down into oxalate and acetate using oxaloacetate acetalhydrolase, or OAH. And then finally, this acetate is channeled back into the cycle using acetate-CoA ligase, or ACS. So in one cycle, we see 2 molecules of carbon dioxide being absorbed and 1 molecule of oxalate being produced. So after choosing this simplified POAP pathway, our next step was finding a host organism that was suitable for testing and screening this pathway. We decided on the JCVI SYN3B minimal cell because of its simplified metabolic framework that ensures that any pathways added wouldn't really interfere with existing metabolic reactions or cause any intermediary side products. Additionally, we think it would be useful to study the metabolomics of this POAP pathway using GCMS to see if there were any consequences of the pathway on the most central native metabolism found in all cells. Since the minimal cell only encodes the genes necessary for cell survival, it would be easier to screen on GCMS because there would be less background noise and it would be relatively straightforward to see what's happening inside the cell. Finally, because the minimal cell's function is essentially to do nothing, and since it freely diffuses acetate, we believe adding this pathway that consumes acetate in a cyclic manner wouldn't really hinder or burden the cells. So that being said, the goal of our project was to insert this 4-step POAP pathway into the JCVI minimal cell. Now, all 4 enzymes combined come out to be around 10 kilobase pairs, which we would then put into the 531 kilobase pair minimal cell. So the first step of our process was to screen the enzymes needed to put into our cells. For this specifically, we wanted to look at the operating temperatures of these enzymes to make sure they were within the range that the minimal cell could survive in. So we decided on these 4 enzymes highlighted here from these organisms, but if you look closely, the P4 enzyme operates at slightly higher temperatures than the minimal cell can handle, which is around 40 degrees Celsius. Additionally, P4 in general is a little bit tricky to use because in nature, it's commonly known for its forward reaction, which is oxidative decarboxylation. Now, if you remember, Sage spoke about this, but so in this process, it uses pyruvate dehydrogenase to break down pyruvate into acetyl-CoA and CO2. However, we want this to be a carbon fixating step, so we want the opposite reaction to occur. So for our purposes, it's great that the minimal cell has not been evolved to include pyruvate dehydrogenase, and using this baseline, having the reverse reaction is beneficial for us. However, the process we want, which is reductive carboxylation, is less thermodynamically favorable So there are a few things we have to consider to make sure P4 works the way we want. So the first is having a reducing agent readily available. And of course, that's easier for us in an in vivo setting because NADH already exists in the cell and can be used for this process. The second step is creating a disequilibrium in the system where we'll take advantage of lactate dehydrogenase, which is naturally occurring in the minimal cell. And this enzyme works at a much faster rate than P4. So what we imagine is that as the acetate is turned into pyruvate, the pyruvate produced immediately gets converted into lactate. And this depletion of pyruvate will favor our reverse reaction and constantly kind of drive the reductive carboxylation of pyruvate. Oh, sorry, acetyl-CoA. And lastly, we want to focus on temperature because, like I mentioned, P4 operates at temperatures higher than 40 degrees. So for that, we decided to use thermal adaptive laboratory evolution to actually evolve our cells to grow at temperatures we wanted. In order to do this, we passaged the cells at a 1:10 ratio through bottleneck temperatures. And so once it was at a bottleneck temperature, we would wait until it had overnight growth, which was monitored through a color change. And then we would bump it up to a new temperature and keep repeating this cycle until we reach a desired level. So if you see on the right here, some of our lines didn't make it, but after 4 months, we got 3 strains that operate at 44.2 degrees Celsius, which was a big achievement for us. So after we dealt with the P4 enzyme, we moved on to our transformation process. Our first step was to order all the primers and genes and to assemble our genes using amplification and crossover PCR. For this process, we actually used the Opentron's OT2 robot, which had a lot of PCR protocols devised, and we used this to help speed things up. And then after we got our genes assembled correctly, we put them into our E. coli and sent them for sequencing to make sure that they were, no mutations, they were correct. And then after that, we transformed them into our mycoplasma, where we used SP4 agar plates with the puromycin resistant genes, and we used a suicide plasmid so that our gene of interest was integrated into the minimal cell landing pad using a Cre-LoxP system. After getting these transformed minimal cell strains containing each of the enzymes separately, we moved on to GCMS analysis. So for GCMS, we went to the Scripps Institution of Oceanography, and we had the help of Dr. Immo Burkhardt at the Moore Lab. So for the first step of GCMS, we need to run standards to see where the peaks of our compounds are. Unfortunately, oxaloacetate and acetate proved difficult to use on the machine using the derivatization protocol created by Dr. Burkhardt on the right. So instead, we decided to focus on the pyruvate steps, which involved the enzymes PYC and P4. These are the carbon fixating enzymes, so we thought it was better to measure the pyruvate perturbations and to at least make sure that, the fixation steps were working correctly. Like I mentioned earlier, P4 produces pyruvate and PYC consumes it. So in the graph here, you can see the peak for the pyruvate standard, and that's the graph we got. We then moved on to run create the metabolic profiles of the untransformed minimal cell, which we would use as a baseline, which you can see on top, and then the P4 transformed cell, which you can see at the bottom. So these were the 2 peaks we got. We decided to look more into lactic acid since we saw a great perturbation in the peak for that. And then over here on the top again is the untransformed minimal cell, and then on the bottom is the minimal cell transformed with PYC. So the data we got was slightly different than what we expected. And so what we did was we converted the data we got from the graphs into area of pyruvate per milligram of cell pellet mass, and then area of lactic acid per milligram of cell pellet mass. On the left, you can see our baseline for pyruvate and lactic acid in the untransformed cell. And then in the middle for P4, we would expect an increase in pyruvate since it's producing pyruvate, but we saw an 18% decrease in pyruvate. However, there was a 48% increase in lactic acid. And we believe this is because, like I mentioned, the state of disequilibrium the cell is in, where as soon as a pyruvate molecule is produced, it's consumed by lactate dehydrogenase relatively fast and produces lactic acid. So we think the P4 enzyme is working the way we want, and it's just that the pyruvate that was produced was converted into lactic acid. So that's why we see that increase there. And then for the PYC strain on the right, we see the trend that we expected, where because it's consuming pyruvate, there's a decrease in both pyruvate area and lactic acid area. So after confirming the function of the carbon fixating steps, we decided to move to the next step of our assembly process, which was to create 2 enzyme designs. So this is something similar to what the authors of the POAP pathway did when they tested in vitro. And their motivation was that it would be easier to test the functionality and optimize half of the pathway at one time. So what we did is we created 2 plasmids. Plasmid A contains ACS and P4, and then plasmid B contains OAH and PYC. Now, for plasmid A, it will consume acetate and produce pyruvate, while plasmid B will do the opposite and consume pyruvate, producing acetate. So we think these halves would be easier to run on GCMS because we would only have to focus on 2 compounds, acetate and pyruvate. And once again, we can hopefully optimize and see what's happening in these 2 halves before putting them together. So how we did this is that we put 2 of the bigger enzymes onto a vector backbone, and then we used the other pair for a gene insert. So in our case, PYC and P4 were put on the vector backbones, and then OAH and ACS were the gene inserts respectively. So for the gene inserts, we had to do a series of PCRs to build the overhang regions successfully. On the left, you can see ACS only required 2 reactions, and we built those up the first time. However, OAH was a little bit more tricky, and it took a couple of tries, but finally we got all 7 PCR steps to work, and we got the 2 fragments of the correct sizes. So we then performed Gibson assembly and sent them for sequencing. And we got confirmation that the enzyme pairs were correctly integrated onto their respective plasmids. After this point, we transformed them into our minimal cell. And now the current stage we're at is that we have 2 strains of the minimal cell containing half of the pathway each. Now, since this is an ongoing project, our next steps would be to perform GCMS analysis and confirm the functionality of the 2 enzyme plasmids in the minimal cell. We would also have to transform the evolved minimal cell, which grows at 40 degrees Celsius, and run that through GCMS to see if possibly the enzymes are working at a faster rate there, or if temperature is helping us or hindering us. It'd be good to quantify that. And then after looking at that, we would want to put both halves of the pathway together and create our 4 enzyme plasmid assembly. We would then integrate this into the minimal cell, and it would definitely be beneficial to do some sort of carbon labeling where we can follow the carbon molecule in the environment and see if it's actually being integrated into the pathway like we'd hope. However, if we want to take this to a larger scale, it's important that we address a few considerations. One of that being is the oxalate buildup in the pathway, which could be toxic to cells. So some ideas we've kind of brainstormed are that there is an organism, there's a bacteria that can naturally break down oxalate into formate and use it for cellular processes. So, that's a potential candidate, or that enzyme could be useful. And then another way is possibly precipitating the oxalate into calcium oxalate. And again, these considerations are more important when looking at what a viable host organism would be, since of course the minimal cell would not be feasible on a large scale due to its expensive media and very specific growth conditions required. So, to conclude, this is in its beginning stages. There's a lot more work to be done, but for everything that we've done so far, I would really like to thank our PI, Dr. Yo Suzuki, and our mentor, Dr. John Glass, along with Dr. Immo Burkhardt for all of the GCMS help, and of course, our student team and our collaborators and external advisors. But thank you everybody for listening, and please let me know if you have any questions.
Very nice, Rridhisha. Questions?
Yeah.
I would have a question. Actually, I think 2 questions. First of all, if you start changing the amount of pyruvate in the cell, wouldn't that kind of cross-react with the PTS, with the phosphotransferase system? Because I remember from the calculations that the Schulten lab did, pyruvate depletion led to the death or the abolishment of metabolic networks. So I wonder, when you have a reaction that brings down pyruvate, that may hinder sugar uptake.
That's definitely, I will look more into that, but I think that would be definitely more important when testing the enzyme halves. But once we put the enzyme cycle together, since pyruvate will be getting produced and consumed, we're hoping the net is kind of the same as what the minimal cell would have. You know, since like that cycle, since it's synthetic and carefully crafted, it would kind of be different than whatever's happening inside the cell. So yeah, we're hoping that since it's producing and consuming pyruvate, you know, definitely we have to look into this more. And I will.
Very short. The second question is: you only have a handful of enzymes. Does it work? Does it work in a test tube? The reaction that you want to install in the mineral cell?
So the POIP pathway was tested in vitro first, and they did have they had to consider a lot more things, but they also had a few advantages, like they could crank up the temperature to, you know, whatever they wanted. So it has been tested in vitro, and you know, the rate I mentioned, 8 nanomoles of carbon dioxide, is what they showed there. And I'm sure that the rate will be slightly less in the cell, but you know, considering other pathways, most of the synthetic pathways have been done in vitro, but they've had a lot of harsher conditions that our cell couldn't survive, and this was the only pathway that kind of came close to what we thought the cell could handle.
Thank you.
Yeah.
I have a question for you, Rridhisha. Very, very nice talk. This is Kim here. I wondered about your adaptation to temperature, that whole series of things. The assumption is that those are mutational, and that you would presume that your enzyme has a higher throughput, if you will. But is there an alternative explanation to that, that it would be more of a lipid survival would be more of a I think the homeoviscous adaptation concept comes in again, where you might have survival and division occurring, which is not really mutational at all. And not that that would make any difference. Well, yeah, I just wonder if you've considered that, and have you, do you plan to check mutational changes in the genome, not just the enzyme gene?
Yeah. So we had sequenced the cells that grew at 44.2 degrees, and we saw, I think we got around 24 mutations. Most of them were relating to the lipoprotein, which we would expect since, you know, having the membrane kind of like more resistant to heat would definitely benefit the cell. But we also saw a few mutations in the sugar transport. And so we have these, we haven't looked at them. This was kind of more of a side project, but definitely we should do a little bit more work in studying what these mutations mean and how they would affect our cells.
Mm-hmm. Thank you.
Other questions for Rridhisha? Rridhisha, as we mentioned yesterday and discussed yesterday in our practice talk, so one of the enzymes in your pathway requires iron sulfide clusters, which there's no reason to think exist in the minimal cell. What are your thoughts about that?
Yeah, so the paper, the authors of the POIP paper, they were looking at coupling the iron sulfide cluster. So P4 contains iron sulfide, and mostly they were kind of looking into coupling that with some sort of reducing agent. They used ferredoxin, and it's mostly so that the reaction works the way we want and it has enough reducing power for the reaction. However, we believe that because the minimal cell already has NADH as a reducing power, the enzyme should still function the way we want, because as long as it kind of has that, it's still, I mean, we still see its functionality, and we'll definitely look more if like the enzyme isn't working the way we expect. But I do believe, John, that you were previously working on iron sulfide clusters, and there's a strain that potentially does have that. So we could definitely look into using that as our host as well.